राजस्थान राजस्थान
हाँ वो इस बार सेंटर डीएमएलटी का है ना, यहाँ पे डीएमएलटी कॉलेज में सेंटर है केमिस्ट्री।
अच्छा अच्छा करो गुड आफ्टरनून can you hear can you hear okay so steps in translation process there are basically three steps in translation initiation elongation and termination. What is translation? It is the synthesis of proteins, that is the chain of amino acids, which are known as polypeptides. You can call it as a second part of central dogma of molecular biology, which says that RNA is synthesized from DNA by the process of transcription, and proteins are synthesized from mRNA by the process of translation. So this is translation is the second part of central dogma. Where does it take place? It takes place in the ribosomes. These ribosomes are found in the cytosome. They are freely present or they may be attached to the endoplasmic reticulum in eukaryotes. These ribosomes, they can read the sequence of the codons on mRNA and tRNA molecules, which transfer this coded sequence in the form of amino acids. So, various enzymes are involved in this process, proteins and enzymes are involved in this process. Basically, translation reads the genetic code in the mRNA. Last lecture, we have seen what is genetic code. So, reading of this genetic code is possible with the help of translation. You can summarize this process into initiation, elongation, and termination. Translation occurs by two main factors, a translator molecule and a substrate molecule. Translator is the molecule which carries out or conducts the translation, while substrate is nothing but the mRNA which is translated into a new protein. You can also call it as a translator desk. So one is the translator, that is tRNA, which will translate this coded information in the form of amino acid, and the second one is the substrate, that is the mRNA, which contains the coded information in the form of genetic code. This translation is guided by ribosomes. So ribosomes, they are made up of ribosomal RNA and proteins. They are also ribozymes which are involved in this particular process. The rRNA or ribosomal RNA has transferase activity. So peptidyl transferase means transfer of or formation of peptide bond between two amino acids. Ribosomes have two subunits of rRNA and proteins. The larger subunit consists of three active sites, E, P, and A: exit site, peptidyl transfer site, and attachment site. These are the important sites where actually the process of uh, protein synthesis or translation takes place. tRNA. A tRNA has anticodon arm and amino acid arm. So anticodon arm, it consists of anticodons, and tRNA has at the other end it has amino acids. Lysine, for example, is coded by the enzyme AAG. So the anticodon of lysine on tRNA will be UUC. Okay. So when this particular AAG codon, it appears on the mRNA, the anticodon UUC on the tRNA will bind to it for a short period of time. When this is bound, there will be recognition of codon and anticodon on the tRNA, and tRNA will release the first amino acid or the lysine amino acid. So these amino acids, as they are released or transported to the ribosomes one by one, they result in the formation of a polypeptide chain. So this polypeptide chain will go on growing till a termination codon is reached. The enzymes and their role in translation. Peptidyl transferase is the main enzyme which is involved in the process of translation. It is found in the ribosomes and it helps in the formation of covalent peptide bond between two adjacent amino acids. So peptidyl transferase is the key enzyme involved in the formation of the peptide bond, CONH bond, a bond between the carboxyl end of one amino acid and amino group of the second amino acid. त्या दोघांमध्ये जे CONH बॉन्ड तयार करतो त्याला आपण म्हणतो पेप्टाइड बॉन्ड, अँड द formation of this peptide bond is catalyzed by the enzyme peptidyl transferase. The enzyme has two substrates. One is the growing peptide chain and another one is the amino acid that is already attached to the chain. It is located on the larger subunit of the ribosome. So the main function of peptidyl transferase is to catalyze the addition of amino acids one by one into the polypeptide chain, where it is present on the larger subunit, in the larger subunit of the ribosome. Peptidyl transferase is made up of RNA and its mechanism is mediated by ribosomal RNA. In prokaryotes, the 23S subunit, note it down somewhere. MCQ मध्ये विचारू शकतात. In prokaryotes, 23S subunit contains peptidyl transfer between the A site and the O site of tRNA. And it is found in eukaryotes in the 28S subunit. Prokaryotes मध्ये हा peptidyl transferase कुठे असतो? 23S subunit of the larger subunit of ribosome. In eukaryotes, it is found in 28S subunit in the larger subunit of ribosome. Next, the mechanism of translation. Translation is initiated when ribosomes, they recognize the initiation codon on mRNA. So here the tRNA will bind and will release the first amino acid. In case of prokaryotes, the first amino acid is N-formyl methionine, and one by one the amino acids will be later on added during the elongation process. The ribosome then shifts its position on mRNA. It is not the mRNA which moves, the ribosome moves from the first position to second position. So the second codon on the mRNA will now be recognized by the anticodon on the tRNA, and it results in the release of the second amino acid, and in this way the elongation process takes place. This elongation will go on continuing till the termination codon or stop codon or nonsense codon is reached. The ribosome will then split into two parts and it will again rejoin when some other mRNA is to be translated. So several proteins are involved in the initiation, elongation, termination factors along with the energy compound GTP. So this GTP will be converted into GDP by releasing 1-phosphate and energy. This is the overall translation process. Now, translation initiation in case of prokaryotes. Protein synthesis initiation takes place by various factors which are known as initiation factors: IF1, IF2, IF3, mRNA, ribosomes, and tRNA. They all form the initiation complex. कोणकोणते initiation factors? IF1, IF2, IF3, mRNA आणि ribosomes आणि tRNA. The smaller subunit of the ribosome upstream on the 5' end of the start site. So ribosome कुठे attach होतो? 5' end mRNAच्या attach होतो, त्याच्या आधी attach होतो आणि नंतर त्याचं स्कॅनिंगचं प्रोसेस सुरू होतं. So it will scan the mRNA in which direction? 5 prime to 3 prime direction. The first codon is usually AUG, which codes for N-formyl methionine. Sometimes it may be GUG or UUG. They may code for valine. So any of this codon should be present to start the process of translation or protein synthesis. So initiation factor, it carries the methionine amino acid which binds to the P site on the ribosome. The synthesis of this first amino acid will be N-formyl methionine. So here tRNA will be known as formyl methyl tRNA. त्याचा tRNA मिथायनिनचा tRNA तो तिथे कॅरी करेल ऑन इट्स अँटीकोडॉना आणि अमायनो ऍसिड अटॅचमेंट साईटला तो मिथायोनिन कॅरी करेल एन फॉर्मेल मिथायोनिन. सो पॉलिपेप्टाइड चेन मध्ये युजली पहिला अमायनो ऍसिड जो असेल तो एन फॉर्मेल मिथायोनिन इन प्रोकॅरियोट्स. There are 3 steps in initiation. Initiation of the binding of mRNA to the smaller subunit of the ribosome, that is 30S subunit, stimulating the initiation factor F3. So binding of mRNA to 30S subunit will first stimulate IF3. This will dissociate the ribosomal subunits into two. Initiation factor IF2 will then bind to GTP and to the initiator amino acid, that is F-methionine tRNA at the P site of the ribosome. The ribosome protein splits the GTP that is bound to IF2. GTP ला अटॅच होतो IF2 म्हणजे एनर्जी रिच कंपाऊंडला अटॅच होतो आणि रायबोसोमल प्रोटीन त्याला स्प्लिट करून जी एनर्जी रिलीज होते दॅट एनर्जी विल हेल्प इन carrying out the assembly of the two ribosomal subunits. Once this is done, IF2 and IF3 will be released. So initiation factors will be released. Let us see the overall process diagrammatically. So initiation, this is the mRNA. So ribosome will attach to this नंतर इथे स्मॉलर सबयुनिट आणि लार्जर सबयुनिट. दीज आर द 3 साइट्स, ए साइट, पी साइट, ई साइट. अटॅचमेंट साइट, पेप्टिडिल ट्रान्सफर साइट, एक्झिट साइट. सगळ्यात पहिल्यांदा tRNA अमायनो ऍसिडला कॅरी करेल आणि mRNA वर येऊन कोडॉन अँटीकोडॉन रेकग्निशन होईल आणि मग अमायनो ऍसिड्स रिलीज होतील इलॉंगेशनच्या प्रोसेसच्या वेळी. सो इलॉंगेशन प्रोसेसच्या वेळी काय दिसते तुम्हाला? वेट. During the process of elongation, you can see, one second. सो इलॉंगेशन मध्ये इथे तुम्हाला दिसते हा पहिला अमायनो ऍसिड होता एन फॉर्मिल मिथायोनिन. आता दुसरा tRNA आलेला आहे, दुसरं कोडॉन अँटीकोडॉन रेकग्निशन होतं आणि सेकंड अमायनो ऍसिड रिलीज झाला. तो सेकंड मग पहिल्याला अटॅच होतो, हा tRNA बाहेर निघून जातो आणि अटॅचमेंट साईटला तिसरा tRNA मॉलिक्यूल येईल. इथे पेप्टिडिल ट्रान्सफर साईटला येईल आणि मग एक्झिट साईटवरून तो बाहेर निघून जाईल इलॉंगेशनच्या प्रोसेसवर. इथे तुम्हाला हे क्लिअरली दिसेल. अटॅचमेंट साईटवरती जेव्हा प्रिव्हियस tRNA जो असेल तो पी साईटला असेल तेव्हा ए साईट फ्री असेल, ए साईटला कोण येईल? नेक्स्ट tRNA येऊन अटॅच होईल आणि ई साईटवर कोण असेल, ज्याचा अमायनो ॲसिड ऑलरेडी रिलीज झालेला आहे. अशा पद्धतीने ही प्रोसेस इलॉंगेट होत जाईल. नेक्स्ट, टर्मिनेशन. आता हे चेन इलॉंगेट झाली आणि लॉंग चेन प्रोटीनची तयार झाली. नंतर टर्मिनेशनच्या फेज टर्मिनेशन फॅक्टर्स जे असतील ते येतील, रिलीज फॅक्टर्स येतील, दोघं रायबोझोमचे सबयुनिट्स सेपरेट होतील, mRNA डिसइंटिग्रेट होऊन जाईल, जो नवीन प्रोटीन तयार झालेला असेल मग तो प्रोटीन सिंथेसिसची प्रोसेस कॅरी आउट करेल. So in this way, termination will be done. So elongation of translation, it involves elongation factors, that is EFTU, EFTS, EFG. The ribosomal function is known to shift one codon at a time, catalyzing the process that takes place at three sites, A site, P site and E site. So first the charged tRNA. The charged tRNA means the tRNA which carries the amino acid from the cytoplasm and brings it to the site of protein synthesis, that is ribosomal complex. So that charged tRNA, when once it reaches the peptidyl transfer site, it will become uncharged by releasing the amino acid, and then it will move away from the exit site. So in prokaryotes, an amino acid is added at least every 0.05 seconds, which means that 200 polypeptide amino acids are translated in 10 seconds. 10 seconds मध्ये जवळजवळ 200 पॉलिपेप्टाइड अमायनो ॲसिड्स तयार झालेली असेल किंवा 200 पॉलिपेप्टाइडची चेन तयार झालेली, 200 अमायनो ॲसिड्सची पॉलिपेप्टाइड चेन तयार झालेली असेल विदिन 10 seconds. The bond created between these amino acids, it requires GTP. In all, you can say there are three distinct steps involved in translation. First, mediation of elongation factor TU in the entry of amino acyl tRNA to the 8 site. Amino acyl tRNA means tRNA attached to the amino acid. So it will first reach the A site. This will be possible because of elongation factor TU. This elongation factor TU binds to GTP, energy compound. So this will activate elongation factor TU GTP complex to bind to the tRNA. Then this GTP will hydrolyze by releasing energy and will be converted to GDP. And it will give the energy for binding the amino acyl tRNA to the A site. Then the elongation factor TU role is over, it will leave, and also tRNA will also start leaving the A site once the amino acid is released. Elongation factor TS, it helps in releasing the elongation factor TU and GDP complex from the ribosomes, and it helps in the formation of elongation factor TU GTP. Okay, so आधी तो बाइंड करतो, नंतर त्याला GTP मध्ये कन्वर्ट करतो. During this translocation process, polypeptide chain on peptidyl tRNA is transferred to amino acyl tRNA. on the A site, and this reaction is catalyzed by peptidyl transferase. So what does peptidyl transferase do? It will help in the transfer of polypeptide chain on peptidyl tRNA to amino acid tRNA at the A site. The ribosomes will then move along the codon in 5' to 3' direction. This is also again mediated by elongation factor G. This step also requires energy in the form of GTP. Uncharged tRNA will be released from the P site, and finally the tRNA will leave from the exit site. Termination of translation takes place when stop codons are reached. Any of the stop codons, UAG, UGA, UAA, any of this nonsense codon, if they are reached, termination takes place. The triplet stop codons are not recognized by tRNA, but they are recognized by release factor RF1, RF2, which are found in the ribosome. So release factor 1 will recognize the termination codon UAA and UAG. Release factor 2, it will recognize termination codon UAA and UGA. The third factor, known as release factor 3, will recognize which will help RF1 and RF2 in their termination process. When peptidyl tRNA from the elongation step arrives at P step, the release factor of the stop codon, it will bind to the A site. म्हणजे जेव्हा लास्ट tRNA जो असेल तो P साईटवर रीच होईल आणि लास्ट अमायनो ऍसिड रिलीज करेल, तेव्हाच रिलीज टर्मिनेशन कोडॉन येऊन जाईल mRNA वरती. कुठल्या साईटला? ए साईटला येऊन जाईल. मग रिलीज फॅक्टर तिकडे बरोबर tRNAच्या ऐवजी तिथे रिलीज फॅक्टर येऊन जाईल आणि तो त्या टर्मिनेशन कोडॉनला ए साईटला रेकग्नाइज करेल. दिस विल हेल्प इन रिलीजिंग दी पॉलिपेप्टाइड चेन फ्रॉम दी पी साईट, कारण दुसरं पुढचं अमायनो ऍसिड येणारच नाही. आपोआप पॉलिपेप्टाइड चेन तिथून सुटून जाईल आणि रायबोझोम्स, 2 subunits of the ribosomes will dissociate with the help of GTP. mRNA will be left out, which will be finally degraded. After many ribosomes have complete translation process, mRNA is degraded. The mRNA will be broken down into ribonucleotides. These ribonucleotides can be further used in the formation of another mRNA molecule during the process of transcription. तेव्हा पण ribonucleotides लागतील आणि हेच ribonucleotides तिथे कामात येऊ शकतात. What is the difference of protein synthesis in pro and eukaryotes? So first thing, the mRNA in case of eukaryotes is monocystonic. So one gene, it codes for one gene of the polypeptide, while mRNA in case of prokaryotes is polysystonic. It can code for several genes. 3 types of RNA polymerase are used in case of eukaryotes for the synthesis of RNA, but only one type of RNA polymerase is used in the synthesis of various types of RNA. In eukaryotes, both subunits are involved, 40S and 60S. Here it involves the 70S ribosome. Transcription and translation take place separately. They do not overlap. But in case of prokaryotes, these two processes are overlapping. Precursor mRNA will be first synthesized in eukaryotes and it will be further modified to form the mature mRNA. तसा mRNA synthesize करायची गरज prokaryotes मध्ये नसते. No special initiator complex is formed in case of eukaryotes, but in case of prokaryotes, methionine tRNA complex is formed. Starting amino acid is methionine in case of eukaryotes. It is N-formyl methionine in case of prokaryotes. In case of eukaryotes, there is only one initiation and termination site. There may be several initiation and termination sites कारण polysystonic आहे. हा मोनोसिस्टॉनिक आहे म्हणून एकच टर्मिनेशन साइट असेल. इथे अनेक असू शकतील. The ribosomal binding site is Kozak sequence that is centered around the starred codon, while the ribosomal binding site in case of mRNA on the mRNA in case of prokaryotes is Shine-Dalgarno sequence, which is located at -10 nucleotide position, that is upstream the start site. Ribosomal binding site in eukaryotes is Kozak sequence, and it is present near the initiation codon. While in case of prokaryotes, this RBS is located in the Shine-Dalgarno sequence, which is located at -10 position of the initiation codon. Several initiation factors are involved in the process of initiation. In case of eukaryotes, elongation factor, ELF, elongation, sorry, eukaryotic initiation factor 2, eukaryotic initiation factor 2AL, 2A1, eukaryotic initiation factor A2, eukaryotic initiation factor A. पण इथे मात्र तीनच फॅक्टर्स इन्व्हॉल्व आहे, इनिशिएशन फॅक्टर 1, 2 and 3 in case of prokaryotes. There are two elongation factors, elongation factor 1 and 2. In case of prokaryotes, they are TU, TS and G. There is only one release factor in case of eukaryotes to recognize the termination codons, and that is eukaryotic release factor, ERF. In case of prokaryotes, there are three release factors, RF1, RF2, RF3. The genetic code may differ in mitochondria and chloroplast. The genetic code is same in every prokaryotic organisms. म्हणजे जेनेटिक कोड मायटोकॉन्ड्रियल DNA आणि क्लोरोप्लास्ट DNA मध्ये व्हॅरी होऊ शकतो, पण तो जेनेटिक कोड प्रोकॅरियोट्स मध्ये सेमच असतो. Translation inhibitors. So first one is puromycin. It's an antibiotic and it is the analog of terminal amino acyl adenosine part of amino acyl tRNA. Amino acyl tRNA चा हा एक पार्ट असतो amino acyl adenosine, which is located at the terminal end, and the structure of this terminal amino acyl adenosine matches with puromycin antibiotic. So it inhibits the protein synthesis by releasing the prokaryotic. It will take the place of this amino acyl adenosine in amino acyl tRNA, and this will inhibit the process of protein synthesis by releasing the prokaryotic polypeptide chain before the synthesis has been completed. छोटासा तुकडा तयार होईल त्याच्या आधीच टर्मिनेशन होऊन जाईल. This puromycin also contains alpha amino group similar to amino acyl tRNA, and this helps in the formation of covalent bond, which forms covalently bound peptide bond with carboxyl group of the growing amino acid. म्हणजे समजा 5-6 अमाइनो ऍसिड सिंथेसाइज झालेले आणि अचानक तिथे प्युरोमायसिन येऊन गेला तर त्याचा अल्फा अमाइनो ग्रुप जो आहे तो त्या ग्रोईंग चेनच्या कार्बोक्सिल ग्रुपला जाऊन अटॅच होऊन जाईल आणि तिथेच टर्मिनेशन होऊन जाईल. सो कंप्लीट polypeptide chain will not be synthesized. Next, streptomycin. So this is a trisaccharide which affects on the binding activity of formyl methionine tRNA, that is initiation tRNA. तो initiating amino acid carry करतो, तो tRNA molecule ला तो bind होऊ शकतो, आणि this helps in preventing the initiation of protein synthesis. Then there are aminoglycoside antibiotics like neomycin, kanamycin, gentamicin, and they interfere with the decoding site in the 16S rRNA of the smaller subunit of ribosome. A very important part, 16S rRNA, which is unique sequence involved in identification, in taxonomy, in classification, आणि त्याच्यावरती हे अमाइनो ग्लायकोसाइड अँटीबायोटिक्स ऍक्ट होऊ शकतो. Chloramphenicol inhibits the activity of peptidyl transferase. Erythromycin blocks translocation by binding to the 50S subunit. Cyclohexamide blocks peptidyl transferase in eukaryotic ribosomes and it is used as a laboratory tool for blocking protein synthesis in eukaryotes. Diphtheria toxin is there. This diphtheria toxin has A fragment which catalyzes the transfer of single side chain of elongation factor 2 and blocks the process of ट्रान्सलोकेशन ऑफ दी ग्रोईंग पॉलिपेप्टाइड चेन म्हणजे त्याच्यावरती अमायनो ॲसिडला ट्रान्सलोकेट होण्यापासून प्रिव्हेंट करतात आणि त्याच्यामुळे पॉलिपेप्टाइड चेन सिंथेसिस इज टर्मिनेटेड बिफोर इट इज कंप्लीटेड ॲज अ होल. नेक्स्ट, ट्रान्सलेशन इन यूकॅरियोट्स. तो खूप काही डिफरन्स नाही आहे, छोटे छोटे पॉईंट्स फक्त तुम्हाला लक्षात ठेवायचे. So same, you'll define it as a process of translating the sequence or coded information in messenger RNA into the amino acids during protein synthesis. So it is a process in which ribosomes in the cytoplasm or endoplasmic reticulum synthesize proteins after the process of transcription, that is formation of RNA from DNA. It may take place in the cytoplasm or it may take place in the endoplasmic reticulum. So this is the structure of ribosome of eukaryotes. A mammalian ribosome, ATS subunits, molecular weight 4.2 into 10 raised to 6 Daltons. 60S and 40S are the two subunits. The 60S subunit has 28S rRNA, which is made up of 4718 ribonucleotides, then 5.8S rRNA made up of 160 nucleotides, 5S rRNA made up of 120 nucleotides, and there are in all 49 proteins in the 60S subunit of the ribosome. The 40S subunit of the ribosome is made up of 18S rRNA and 33 proteins. 18S rRNA, it is made up of 1874 nucleotides. So this is the basic structure of a mammalian ribosome. Chair model. खुर्ची सारखं असतं असं. These ribosomes usually are present separately. These two subunits are separately and they are made up of mainly proteins and rRNA. 80S and 70S in case of prokaryotes. These subunits, they will form one particular entire ribosome when there is need of protein synthesis or when they bind to the mRNA near the 5' end. When they bind to the mRNA, ribosome reads the nucleotide sequence from 5' to 3' direction. And it will synthesize the corresponding proteins from these amino acids in N-terminal amino terminal and C-terminal, that is carboxy terminal direction. So it reads the mRNA in 5' direction, it synthesizes the amino acid chain from amino to carboxy terminal. Ribosomes are located in the cytoplasm. They are freely floating in the cytoplasm or they may be halting or attached to the endoplasmic reticulum. They are the main sites for protein translation. Each prokaryotic ribosome, it has three binding sites. The amino acyl binding site, that is A site, which is essential for elongation or it is essential for binding the incoming amino acid tRNA. The peptidyl transfer site or peptidyl tRNA binding site where tRNA links to the growing polypeptide chain, and exit site or E site is the binding site for tRNA following its role in translation and release from the ribosome. All these three A, P and E sites are formed by RNA molecules in the ribosome. You can see the mechanism of translation here in this particular diagram. इनिशिएशन प्रोसेस. इनिशिएशनमध्ये डायरेक्टली पेप्टिडिल साईटला जातो tRNA. पहिला अमायनो ॲसिड रिलीज करेल. इलॉंगेशनच्या वेळी मग तो पॅटर्न फॉलो करेल ए, पी आणि ई साईटचा. सो पी साईटवरून पहिला अमायनो ॲसिड रिलीज होईल तेव्हा ए साईट दुसऱ्या अमायनो असाईल tRNA ऑक्युपाईड असेल. वन बाय वन अमायनो ॲसिड्स रिलीज होत जातील. ई साइट will be the exit site, so that will be recognized by the termination, that will be recognized by the release factors. The termination codon will be recognized by the release factor. So site of action प्रत्येकाची त्यांनी दाखवलेली आहे. Mechanism of translation in eukaryotes. ऑलमोस्ट सेम, काही थोडे थोडे डिफरन्सेस आहेत. रायबोझोमल युनिट्समध्ये डिफरन्स आहे. तिकडे 70S मेडअप ऑफ 30 आणि 50S, इकडे 80S मेडअप ऑफ 40 अँड 60S. युकॅरियोटिक रायबोझोमल सबयुनिट्सचं कॉम्पोजिशन थोडंसं कॉम्प्लेक्स असतं प्रोकॅरियोट्सपेक्षा. mRNA जो असतो तो मोनोसिस्ट्रॉनिक असतो. नंतर पोस्ट ट्रान्सलेशन क्लिवेज रिॲक्शन्स तिथे होत असतात. इट रिक्वायर्स 9 eukaryotic initiation factors, that is eukaryotic IFs initiation factors. फक्त 3 लागतात प्रोकॅरियोट्समध्ये, युकॅरियोट्समध्ये 9 लागतात. The initiating amino acid is not N-formyl methionine but only methionine. Special tRNA is required for initiation in case of prokaryotes. Shine-Dalgarno sequence is present in prokaryotes. Kozak sequence is present in eukaryotes. Eukaryotic mRNA do not contain Shine-Dalgarno sequence. A 40S ribosomal subunit attaches to the 5' end of the mRNA, and that region is known as the Kozak sequence. And then it will move along the mRNA in 5' to 3' direction, which is known as the scanning of mRNA. Let us see these stages in detail. So the first step is the pre-initiation complex formation, consisting of the 40S subunit of the ribosome, methionine tRNA, eukaryotic initiation factor 2 and GTP. This is the pre-initiation complex which binds to the 5' end of the mRNA. This step also requires initiation factor 4F and elongation factor, initiation factor 3. This is also known as cap binding complex. Then initiation factor 4F complex consists of initiation factor 4A, 4E and 4G. Initiation factor 4E first binds to the 5' region of the cap, while the initiation factor 4G interacts with the poly A tail binding to the poly A by binding to the poly A tail. लक्षात ठेवा, प्री इनिशिएशन कॉम्प्लेक्समध्ये इनिशिएशन फॅक्टर 4F आहे आणि इनिशिएशन फॅक्टर 3 आहे. आता 4F कॉम्प्लेक्स जो आहे तो कशाने बनलेला आहे? 4A, 4E आणि 4G. त्यातला 4E जो आहे तो कॅपला अटॅच होतो. 4G जो आहे तो टेलला अटॅच होतो. mRNA ला कॅप आणि टेल असतं eukaryotes मध्ये. Initiation factor 4A is ATP dependent RNA helicase that will unwind any secondary structure if it is formed in mRNA. कारण mRNA मध्ये काय पॉसिबिलिटी असते की कॉम्प्लिमेंटरी बेस पेअर्स जिथे असतील तिथे ते एक दुसऱ्याबरोबर हायड्रोजन बॉन्डिंग करून लूप स्ट्रक्चर्स तयार करतात. तर ते लूप स्ट्रक्चर्स चुकून जर तयार झालेले असतील, कारण eukaryotic mRNA or genes will be very large, सो देर आर चान्सेस ऑफ फॉर्मेशन ऑफ लूप स्ट्रक्चर्स. त्या लूप स्ट्रक्चरला ओपन अप करायचं काम हे 4A इलॉंगेशन इनिशिएशन फॅक्टर करेल. Then it will move along the mRNA in 5 prime to 3 prime direction. That process is already known as scanning. The 5 prime untranslated region of eukaryotic mRNA varies in length. It may be hundreds of nucleotides long, and it consists of secondary structures called hairpin loops. The 5 prime untranslated region. 5 प्राइम्स एंडला अनट्रान्सलेटेड रिजन असू शकतो युकॅरियोटिक mRNA मध्ये, आणि तो हेअरपिन स्ट्रक्चर तयार करू शकतो. दीज सेकंडरी स्ट्रक्चर्स आर रिमूव्ड बाय इनिशिएशन फॅक्टर्स ऑफ द स्कॅनिंग कॉम्प्लेक्स. वरती आपण पाहिलं. The initiation codon is recognizable because it often contains a short sequence called the Kozak consensus, that is 5 prime ACC AU GG 3 prime sequence. The initiation codon and termination codon madhe identification is nothing but this particular sequence. This is known as the Kozak consensus sequence in case of eukaryotes and Shine-Dalgarno sequence in case of prokaryotes. Once the scanning complex is exactly positioned on the initiation codon, the 60H larger subunit binds to form ATS. It will bind to the 40S and it will form the ATS initiation complex, and this particular step requires GTP, energy in the form of GTP. Elongation is carried out by eukaryotic elongation factors 1A, 1B, and elongation factor 2. These are TU, TS, and G in case of prokaryotes. एवढाच फरक आहे. दोघं इथे सुद्धा 3 आहेत आणि तिघांचं जे रोल आहे तेही तसंच मॅच होत असतं. इलॉंगेशन फॅक्टर 1A, 1B, and 2. At the end of initiation, mRNA is positioned so that the next codon, elongation पहिला codon तर झालेला आहे, that is methionine codon recognized झालेला आहे. So the next codon can be translated during the elongation step of protein synthesis. The initiator tRNA will now take the position of the P site and A site will be free. This A site will now be occupied by the second amino acid tRNA as per the second codon on the mRNA. So here this elongation process will go on continuing. Addition of amino acids will take place, and an nascent polypeptide chain is formed in 3-step microcycle. So what is this 3-step microcycle? Positioning the correct amino acid tRNA at the A site of the ribosome, formation of peptide bond between the first amino acid and the second amino acid, shifting of mRNA by one codon so as to reach the third codon on the mRNA. The 3 steps आहेत. Exact positioning at the attachment site, then movement to the peptidyl transfer site, recognition होईल, अमायनो ऍसिड्स रिलीज होतील आणि पहिला अमायनो ऍसिड आणि दुसऱ्या अमायनो ऍसिड मध्ये पेप्टाइड बॉन्ड तयार होईल. रायबोझोम आता पुढे सरकेल आणि तिसऱ्या कोडॉनला जाऊन एक्झॅक्टली पोझिशन त्याची घेईल. During elongation in bacteria, deacylated tRNA in the P site moves to the exit site. ज्याच्यातनं अमायनो ऍसिड रिलीज झालेला आहे असा tRNA एक्झिट साइटला जाईल. In case of eukaryotes as well as in bacteria. In contrast, although the situation is still not completely clear in eukaryotes, deacylated tRNA appears to be रिमूव्ड फ्रॉम दी रायबोसोम्स. म्हणजे प्रोकॅरियोट्स मध्ये मात्र ही सिच्युएशन आपल्याला क्लिअरली दिसते की रायबोझोमच्या ई साईटला तो tRNA जातो आणि तिथून तो एक्झिट होतो. पण युकॅरियोट्स मध्ये हे मात्र क्लिअरली अजूनपर्यंत लक्षात आलेलं नाही. तरी पण तो तिथून इजेक्ट होतो, रिमूव्ह होतं, हे मात्र तितकंच करेक्ट. नेक्स्ट इज टर्मिनेशन. Termination depends upon release factors, and these release factors are RF1 only in case of eukaryotes. So RF1 will recognize all the 3 termination codons UAA, UAG, and UGA, and release factor 3 will support release factor 1 for this process. Then the ribosome assembly will break and the polypeptide chain will be released. Next is cytoplasmic polyadenylation, CPE. The cytoplasmic polyadenylation element, it is nothing but a sequence found in 3' untranslated region of messenger RNA. Ribosome moves from 5' to 3' direction, पण 3' एंडला टर्मिनेशन कोडॉन आल्यानंतर पण काही असा रिजन असतो which is not translated. The most common cytoplasmic polyadenylation sequence is UUUUUAU. Binding of cytoplasmic polyadenylation element protein CPEB to this region promotes the extension of the existing poly A tail. आता काय होतं की mRNA in eukaryotes consists of poly A tail आणि तेवढा पोर्शन काही ट्रान्सलेट होत नाही कारण त्याच्या आधी टर्मिनेशन कोडॉन घेऊन पोचलेला असतो. तर अशी जी अनट्रान्सलेटेड रिजन असतो, ओके, त्याला सायटोप्लाझमिक पॉलीएडिनिलेशन एलिमेंट बाइंड होतो किंवा प्रोटीन बाइंड होतो आणि त्याच्यामुळे काय होतं की ती टेल अजून लांब एक्सटेंड होत जाते. सो दिस हेल्प्स इन द एक्सटेंशन ऑफ द एक्झिस्टिंग पॉली ए टेल. This elongation occurs after mRNA has been exported from the nucleus to the cytoplasm. ही एक्सटेन्शनची प्रोसेस जी आहे ही केव्हा होते? जेव्हा सायटोप्लाझममध्ये mRNA ट्रान्सपोर्ट झालेला असतो न्यूक्लियसमधून थ्रू दी न्यूक्लिअर मेम्ब्रेन बाय द फॉर्मेशन ऑफ अ न्यूक्लिअर पोर. mRNA न्यूक्लिअर पोर मधून बाहेर निघतो न्यूक्लियसमधून आणि सायटोप्लाझममध्ये पोचतो, त्याच वेळी ही प्रोसेस झालेली असते. A longer poly A tail attracts more cytoplasmic polyadenine binding proteins. जास्तीत जास्त प्रोटीन्स मग अशा लॉंग पॉली ए टेलला अट्रॅक्ट होतात and they interact with several other cytoplasmic proteins that encourages mRNA and ribosome to associate. So this extension of poly A tail will attract many poly ABP proteins, and these proteins will help in the interaction between the ribosome and the mRNA. So it is believed that poly A tailचं जे एक्सटेन्शन होणं किंवा लेंथनिंग प्रोसेस जी आहे ती ट्रान्सलेशन प्रोसेसची एफिशियन्सी वाढवत असतं. आणि कितीने ती टेल लांब होते तर 40 बेस पासून 150 बेस पर्यंत ए ए ए ए ची सिक्वेन्स एक्सटेंड होऊ शकते इन केस ऑफ यूकॅरियोट्स. Cytoplasmic polyadenylation can be distinguished from न्यूक्लिअर पॉलीएडिनिलेशन. ह्या दोन वेगळ्या प्रोसेसेस आहेत. सायटोप्लाझमिक पॉलीएडिनिलेशन occurs in the cytoplasm in specific mRNAs, while nuclear polyadenylation it affects almost all mRNAs. न्यूक्लियसमध्येच ए टेल जी होते ही सगळ्या mRNA मध्ये होऊन जाते, पण सायटोप्लाझम मध्ये गेल्यानंतर मग ती ए टेल एक्सटेंड होते, ती काही mRNA मध्ये होते, काहींमध्ये होत नाही. Among other functions, a prominent role of cytoplasmic polyadenylation element is identified in oogenesis, spermatogenesis, mitosis, and new growth of new synapses. तर ही प्रोसेस, ह्याच प्रोसेसेस ज्या आहेत त्याच्यामध्ये मात्र पॉली ए टेलचे एक्सटेन्शन दिसून येतं. The role of CP was first characterized in xenopus oocytes and embryos. पण सगळ्या सोमॅटिक सेल्समध्ये मात्र त्याचा रोल अजूनपर्यंत माहिती पडलेला नाहीये. पॉली ए टेल का एक्सटेंड होतं, सायटोप्लाझमिक पॉलीएडिनिलेशन का होत जातं, जवळजवळ 150 बेसिस पॉली ए बेसिस का तिथे ॲडअप होतात, तर इट इज believed to be increasing the efficiency of translation process. That is the only role studied to date. And it was first observed that this type of polyadenylation is not found in all mRNAs. It is found only in some mRNAs, and these mRNAs will be involved in the process of oogenesis, spermatogenesis, mitosis, म्हणजे डिव्हिजन मध्ये जे इन्व्हॉल्ड आहेत किंवा रिप्रोडक्टिव्ह सायकल्समध्ये किंवा एम्ब्रियॉनिक डेव्हलपमेंटमध्ये जे इन्व्हॉल्ड आहेत त्या प्रोसेसेसमध्ये ही प्रोसेस सायटोप्लाझमिक पॉलीएडेनिलेशन जास्त एफिशियंटली दिसून येते रादर दॅन इन सोमॅटिक सेल्स. The eukaryotic, cytoplasmic polyadenylation, CCPE. It is again cytoplasmic polyadenylation and embryonic cytoplasmic polyadenylation. It is found in the embryos and it's known as ECPE. आणि त्याच्यामध्ये जी सिक्वेन्स ऍड होते ते UUU सिक्वेन्स. While the sequence of C polyadenylation is CCCCC region and is मध्ये कुठेतरी U regions. All of these CPEs have in common their effectiveness in promoting the extension of poly A tail depending upon their proximity to the poly A signal. CPs can cause translation repression if two CP sequences are located within 50 nucleotides of each other within the 3' UTR, untranslated region. UTR is nothing but untranslated region. The 3' एंडला जो untranslated region आहे तिथे जर दोन सायटोप्लाझमिक पॉलीएडेनिलेशन एलिमेंट्स 50 न्यूक्लिओटाइड्सच्या जवळ असतील तर मग ते ट्रान्सलेशनची प्रोसेस थांबवू शकतात. म्हणून सायटोप्लाझमिक पॉलीएडेनिलेशन एलिमेंट्स शुड बी फार अवे फ्रॉम इच अदर. 50 न्यूक्लिओटाइड्सपेक्षा जास्त त्याच्यामध्ये डिस्टन्स हवं आणि हे एलिमेंट्स जर जवळ आले तर ट्रान्सलेशनची प्रोसेस affect होऊ शकते. The highest amount of repression are seen when two CPs are 10 to 12 nucleotides close to each other. If the CP, cytoplasmic polyadenylation element, has a non-consensus sequence, a nearby pumilio binding element is necessary for translation activity. नॉन कॉन्सेन्सस सिक्वेन्स जर त्याच्यात प्रेझेंट असेल तर मग एक वेगळा एलिमेंट लागत असतो ट्रान्सलेशनची प्रोसेस ॲक्टिवेट करायला अँड दॅट इज नोन ॲज प्युमिलिओ बाइंडिंग एलिमेंट. आणि कॉन्सेन्सस सिक्वेन्स जर त्याच्यात प्रेझेंट असेल तर प्रेझेन्स ऑफ पीबी कॅन डबल द रिझल्टिंग ट्रान्सलेशन ॲक्टिव्हिटी. तर मग ही प्युमिलिओ बाइंडिंग एलिमेंट जी आहे ती ट्रान्सलेशनची प्रोसेस फक्त ॲक्टिवेट करणार नाही पण त्याला डबल efficiency activate करेल. CP is not only the cis acting element to regulate 3' UTR processing as alternative polyadenylation signals, microRNA target sites and AU rich elements. They have all the role in determining the length of फक्त CPEच नाहीये, सायटो प्लाझमिक पॉलीएडेनिलेशन एलिमेंट फक्त हे काम करत नसतो पण देर आर मायक्रो RNAs ऑल्सो किंवा अनट्रान्सलेटेड रिजन्स मध्ये काही असे रिजन्स आहेत AU रिच रिजन्स रिच एलिमेंट्स हे सुद्धा त्याच्यात पॉली ए टेल का किती आणि कुठे एक्सटेंड व्हावी याच्यासाठी रिस्पॉन्सिबल असतात. So this cytoplasmic polyadenylation, it is not a well studied अजून पण त्याच्यावरती खूप सारं रिसर्च होतोय पण एकच आहे की अशा एक्सटेन्शनमुळे ट्रान्सलेशनची प्रोसेस एफिशियंटली होते आणि असं एक्सटेन्शन पॉलीएटेलचं जे आहे ते का होतं तर बिकॉज कुठे होते एस्पेशली इन केस ऑफ एम्ब्रियॉनिक सेल्स, इन केस ऑफ oogenesis, मायटॉसिस म्हणजे डिव्हिजन सायकल्स जिथे जिथे होत असतील तिथे त्या सेल्समध्ये ही प्रोसेस स्ट्रॉंगली दिसून येते रादर दॅन इन केस ऑफ ओनली सोमॅटिक cells. So we'll stop here, continue it in the next lecture. We'll finish translation by Saturday आणि next week मध्ये मग आपल्याला fourth unit सुरू करायचं आहे. So is it clear? साधा सिंपल प्रोसेस आहे. याच्यात फक्त हे जे नवीन नवीन आहेत तेच महत्त्वाचे आहेत. बाकी तुम्ही हे TY ला सुद्धा स्टडी केलेलं आहे. काही डिफिकल्टी आहे? और डिड यू अंडरस्टँड? समजलं? झोपले, नुसते गुड आफ्टरनून करतात आणि झोपून जातात. मनीषा, हर्षाली, कविता, हर्षला, हर्षाली नाही, हर्षला पण आहे आणि हर्षाली पण आहे. व्हेरी गुड. ममता, शिवानी, समजलं का? हो पण म्हणत नाही, नाही पण म्हणत नाही. म्हणजे काहीच झालं नाही, समजलं पण नाही आणि नाही समजलं पण नाही. जागृती, भाविनी, आता धड 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 धड, भारती. ओके, so let us stop here. We'll continue it tomorrow. Thanks for your patient listening.